behalf of Histopathology Laboratory, National Hospital, Kandy, I warmly welcome you for the second day of Molecular Diagnostic Workshop. We'll start with lectures in the morning.
Ayubowan, everybody. It's a pleasure to be here. And at the outset, I would like to thank Dr. Palita and the whole organizing team for having us here in this beautiful country of yours. As they say, correct me if I'm wrong, as they say, Bahuma Shruti, right? So thank you for having us here. So today I'll have about three talks. So what I have done is I've kind of amalgamated these talks in such a way that there is a continuum. I've taken some liberty to play with the headings, and I hope I'm able to send off the basic message what I want to give in the talk. Okay. Yeah. So today in diagnostic laboratories, when you have a tumor or when you have a neoplasm, you have to characterize. How do you go about classifying it or characterizing a tumor or a tumor sample that comes to the laboratory? Of course, you do cytomorphology. So if there is a history of myeloproliferative neoplasm, we call MPN in blast phase, and history of MDS, then we call MDS related changes. But things changed later. I'll just skip this. That in the in the 2022, what has happened is in 2016 and later, patients with recurrent cytogenetic abnormalities, right? We're called AML with recurrent genetic abnormalities, and these patients with these genetic abnormalities were called acute leukemia, irrespective of the blast percentage. For those who are not from pathology background, blast is nothing but the tumor cell in the bone marrow. Okay, it's called a blast. So when you do a bone marrow of a patient with suspected acute leukemia, you count the number of blasts.
Good morning, everyone. So the third talk in my series is basics of NGS, that is next generation sequencing. I would be trying to keep it as simple as possible, but since it's a next generation sequencing, it always tries to be a complex topic to go through. So this is time efficient. We could run maximum number of samples with this. It was cost effective because more number of samples could be run, and 95% genome coverage, including telomeres and centromeres, could be done with this. We called this as poly sequencing, and later on it was called as high throughput or massive parallel sequencing. And as George showed it in 2005, there was a boom, and people started using next generation sequencing for each and everything in their lab. And as you can see, after 2005, the use of next generation sequencing as well as the output of base pairs increased. Nucleotide goes in. If it gets incorporated, pH change happens. So machine knows that the T nucleotide is gone and pH change has happened. So a T nucleotide is. Then G goes, then C goes. This cycle is repeated again. A, T, G, C. A, T, G, C. This cycle repeats, and that is how we get a fragment of DNA nucleotide. That is the actual sequence that was present in the original DNA fragment before the clonal amplification happened. So basically there is something called as a chip that is there in Ion Torrent platform. This chip has this, is not the word to be used, it is variant the word to be used. Change in genetic material which may involve only a single base or large segment of chromosome is actually called a variant. It may be caused due to chemical, radiation, or through inherent error of DNA replication or repair mechanisms. What is polymorphism? Polymorphism is basically a variant which is a genetic change which is found at a frequency of more than 1% within the general population of that particular region. It may produce a phenotype change or it may not produce a phenotype change. Even if it produces a phenotype change, it might not be an harmful change. Like if I say some people have black eyes, Sir has blue eyes. So that's that is used to actually document this particular variant. This is like the state or city within the country that you are living. It's like a country, state or city that is there. Then you go down to the coding region, which is where actually the change is located. This is like the street address, okay? That the house is located at this particular street or the institute is located at this particular street. And finally is the protein change, which actually is like how does the house look. So if you want to give your address, you would be saying ki, my house is located over here, it's a blue color bungalow, a red color bungalow located on this particular street, in this particular city. And if you're going towards the actual Google Maps or the GPS maps, it's basically the latitude and longitude based on which it gives you the actual address that is there. So this is something like
Manjula, consultant histopathologist, District General Hospital, Munavagala, to hand over the token of appreciation from the College of Pathology, Sri Lanka. Parihar to deliver his next talk.
My second talk was fish in solid tumors and third was molecular in solid. So I've taken some liberty and I have done fish and molecular in solid tumor as one talk so that it is much more integrated and easier for everybody to follow. Meanwhile, I thought I introduced another talk here. I thought I'll talk about certain basics of fish, right? And I'll give you our experience as how we built upon a national trial in pediatric leukemia just by using fish in order to risk stratify these patients. So, because a lot of the basics of molecular anyhow we've been talking about, but I'm gonna touch on some of these aspects and we will and talk a little bit of quality control as we go ahead, right? So I'm sure, yeah, all of you know what is fish, okay? Fish, as I said, is a simple technique wherein you have sequences that are complementary to the sequences you want to study, and these sequences have fluorescent probes attached to it. So basically, you can visualize it. You know, I really love this because you are looking at genes through the microscope with your own eyes, right? And it's very exciting. And there are a lot of subtle in embedded tissue. It's always a challenge. A lot of pre-analytical factors are involved. I'll talk about it as we go. We have fished on cytology smears. So you do an FNAC, you have a smear, you can fish on those. You can make cell blocks. So if you're making cell blocks here, you can actually fish on cell blocks, and you can do imprint smears. So you know what we do is there's this lymphoma called the Burkitt's lymphoma, which is characterized by c-MYC gene rearrangements. So whenever we have a patient of Burkitt lymphoma, what we do is the moment the patient has a lymph node biopsy is done, we take the biopsy, we touch it on the slide and make an imprint, right? And even before the histopathologists have seen it, and there is a strong suspicion of lymphoma, on those imprint smears we do the c-MYC break apart fish, and within 4 hours, we are able to give the diagnosis that it is a c-MYC rearranged lymphoma. So it's a very simple technique. Take the tissue, put an imprint on the slide, just do the fish, and of course, I'm not saying morphology is not. I have been a histopathologist, I think in my last birth now. It's been many years I've been doing molecular. That was all, of course, my first love. So you can actually innovate at your local level in order to give better diagnosis and timely diagnosis. So the liquid fish, the slide preparation, your slide has to be clean. You prepare your slides, you do buffer treatment. There is a process of dehydration. If you have any amount of any amount of say water, you will get a very hazy kind of a fish, and you add the probe, then you denature it at 73 degrees centigrade for 5 minutes if you have a thermobrite. If you do not have the thermobrite. Now coming to the solid tumor, As I said, solid tumor is really challenging. There is a lot of issue about the small tissue we have, right? So you have a small biopsy. Now on that biopsy, you have to do histopathology. You have to do immunohistochemistry. You want to do FISH. You want to do molecular pathology. How do you do that? So these were the guidelines. So previously what was done is you do first H&E and then you say okay. Then time of filtration. Over fixed tissues also will cause a problem. Your sections have to be thick, right? So you always need to have a histopathologist who always, actually threatens my technologist how much more thin sections you want. So because of the thick section, you will not be able to get the plane of the signals, and you get multiple planes. It will be difficult for you to start to deparaffinize and rehydrate them. So you remove the paraffin wax, put them in xylene, 96% alcohol, 70% alcohol, and then rehydrate them. You put in a little bit of a heat pre-treatment which digests the methylene, and reduces the autofluorescence. So you put in 0.2 normal HCl, the sodium isothiocyanate, and then you put the enzyme. Okay, now the enzyme here is required to break the protein bonds so that your fish probes are able to go. Now this is a very important step. For example, if you have a solid tumor with a lot of desmoplasia, a lot of fibrous tissue, I'll give an example. Brain tissues are very difficult to fish because there's so much of protein and fat in there, right? Hello everyone. So it's a difficult task post lunch. I usually say, people after lunch undergo apoptosis, right? Automatic ptosis of the eyes. Okay. I'll try to see there is no apoptosis here. Okay. So when you look at the solid tumor assessment in pathology, right? Of course, once we have the tissue, we have histology, we do IHC stain and then IHC, and after that, once you have the diagnosis, and this is where your fish and molecular techniques come into play, and that is when you start picking out you make a choice as to what molecular test has to be done. I've covered this slide. Then we also fish it because ROS, you can have activated macrophages showing ROS IHC positivity and can lead to confusion. So ROS IHC positives we fish anyways. ALK, if it is equivocal, then we fish to rule out gene rearrangement. So again, there are criteria to it. This is by the International Association of Staging of Lung Cancer 2017. Cutoff is 15%. First reader will rate 50 tumor cells. More than 50% is positive. 10 to 15 equivocal. Second reader, then first and second 100, and then you do this. So just follow this criteria, and you will have positive. Because please remember, there is on section sometimes it is very difficult.
For your interesting lecture. Now I invite Dr. Priyangi Amarbandu to hand over the token of appreciation from the College of Pathology, Sri Lanka, to Dr. Malik.
Thank you, madam. A heartfelt thank you extend to all the attendees who graced the molecular diagnostics workshop with their presence, including our esteemed guest speakers from India, participants from National Hospital of Kandy, as well as representatives from various hospitals and universities. A special thank you also goes to the Prime Care Private Limited and CT Sri Lanka Alliance Club for their generous support and collaboration in making this workshop a success. Your dedication and participation has truly made a difference in enriching our event. Thank you immensely for your unwavering support and commitment to advancing the field of molecular diagnostics. Thank you.
Snack outside the auditorium. After having that, we can start the practical session starting from group E to H. Group E will start first.
bottom level is a kind of a vast amount of information is there, vast amount of things to understand, and this will help us to have more precise targets in treating and understanding the disease processes as well. So let us look into the basics of molecular genetics and cytogenetics. I'm just going to give you an overview in the very first introduction here. I think talking about the cytogenetics first, I got right. I went through several definitions also, but this simple definition I thought would be Zira, where people were looking into the molecules. So their technology was good enough to look into study the molecules, and so molecular biology was invented, and molecular biology has invaded into genetics. Changes will help the modification of the expression by suppressing the expression or enhancing the expression, right? So these are something that is outside the DNA or the chromosome material, but these have a bigger effect in causing diseases or when we are looking at the structure function to explain the function, because sometimes we will have the gene talking about a lot of things to patients, giving information, but we are not looking at this counseling part. So even before you do a genetic testing, you have to counsel them, and after that you have to counsel them. There you have to monitor them, because you are dealing with a kind of a base level set of information, so which may be transferable from generation to generation, which may be transferable within the family members. So all
lectures, and all are connected to each other, and basics of PCR is the first thing that I would be starting. So at core I am a pathologist with subspecialization in oncopathology, hematopathology, and molecular genetics. And the outline of talk that is there is basically how I learned my molecular genetics post MD pathology from various institutes that I have been. I learned about basic biology that is there, which was discussed previously. How PCRs are done, components of PCRs, how PCRs techniques are just a platform to see how this central dogma can be utilized in a different way. Another important process is DNA replication, wherein the genetic information is transformed by one cell or mother cell into the daughter cells. And this process of DNA replication is very important because this is the process that actually happens in the PCR. So if you see, there are primers that, firstly, the double-stranded DNA breaks into individual two strands, the primers get attached to it, the polymerase comes in, DNA polymerase, and it starts extending the chain of nucleotides from 5' to 3' end, and later on from the single strand of DNA you have two strands of DNA which further get processed down by mitosis into the daughter cells. What is PCR? It's a simple technique that results in exponential amplification of any region or selected target region of the DNA. PCR works in a similar way to DNA replication, which is a natural process which happens in each and every cell in the body as the mother cell divides to two daughter cells. PCR amplifies DNA, it makes lots and lots of copies of few copies of DNA, and it can copy different lengths of DNA. However, it can't copy the whole length of the DNA that we have in our body. Okay? It's the short segments of DNA that can be amplified by PCR. This is a famous picture wherein Kary Mullis, who discovered the technique of PCR, received Nobel Prize in 1993. The technique of PCR was actually discovered in 1983 when Kary Mullis was traveling through a desert area wherein we saw hot springs wherein algae, Thermophilus aquaticus was growing, and he was famished to see it's in hot water, how is this algae growing and how is it replicating. So he took that algae, he extracted the enzyme DNA polymerase from that, and that DNA polymerase was the turning point in history, I would say, and that is actually the process of PCR that routinely we do in our molecular genetics. The birth of that process took place at that time, and later on multiple modifications of this process of PCR has happened. And there goes denaturation. What is denaturation? Basically, a single strand double helix of the DNA gets converted into single strand. The bonds, the hydrogen bonds between the purines, pyrimidines break and they separate out. And the strands of the DNA separated out undergo a process called as annealing, wherein the primers bind to the segments of the DNA that needs to be amplified, and then there is something called as extension, wherein the actual enzyme, which is the horsepower behind the PCR technique, comes in and starts putting in nucleotide to form a complementary chain of DNA. And PCR basically means polymerase chain reaction. So there is a chain reaction happening. So now the steps are repeated continuously for a number of cycles. This can range from 30, 40, 50, 60, 70 as per your requirement. So basically 3 steps happening continuously in PCR, which are basically denaturation. I'm sorry, there was the temperatures that were over here, these are not seen over here. So denaturation basically happens at a temperature of 95 degrees, wherein the strand breaks it. Annealing, that is primer binding, generally happens at a temperature of 55 to 60 degrees, and then extension happens at a temperature of somewhere of 60 degrees to 65 degrees. So basically PCR is a play of temperature. You increase the temperature, you decrease the temperature from 95 to 55, then again increase it to 65. One cycle is done, repeat the cycle, denaturation happening, and after 20 to 40 cycles you will get a number of amplified copies of your DNA target region that you have amplified. So first cycle starts with a single strand of the DNA, and in first cycle one strand of the DNA is replicated into two strands of the DNA. The product, lower concentration will not give you any amplification. The oligonucleotide primers need to be added in a particular concentration. The DNA template also needs to be added in particular concentration. So polymerase, basically it's a thermostable polymerase. Thermus aquaticus is the algae from which this polymerase was first developed. Now there are multiple polymerases available: Taq polymerase, Wnt polymerase, FUFA polymerase. The half-life of any polymerase is somewhere around 40 minutes. The Taq polymerase, which is commonly used. One of the most important thing that we need to know is the fidelity. Basically, the Taq polymerase is a notorious enzyme. What it does is sometimes while copying, it can introduce its ramps the temperature. It is called as the ramping speed, and different thermocyclers have different ramping speeds. So you can choose whichever is best, which is valuable, cost effective for your setting, and go ahead with that. Nothing major to choose. Now, we have put all the components and we have seen that one copy of DNA, a double-stranded DNA, can go up to 1 billion copies. That's really true. But what is the actual end product of a PCR? So, if I am supposedly putting 10 copies, what would be the product? It's not going to be in billions. The reason is that's the theoretical yield of a PCR. The actual yield of PCR is less. The reason being, we have given a limited components, we have given limited number of primers, we have given limited number of DNTPs. These all are getting utilized. So the theoretical yield might be in billions, but the actual yield is somewhere less. The end product basically is calculated as 2 raised to n plus 1, where n is the number of cycles, 35, 36. So this is just to show you that it's not a billions, but it comes down to somewhere around millions, but that is also sufficient enough in a PCR cycle. Now coming to the main question, how is the amplified product visualized? I said DNA is not seen. So how do you see DNA or the amplified product? Basically, gel electrophoresis is something that is done. It's a conventional technique that is used for looking at whether your PCR has worked and the product of interest has amplified. It's a simple technique wherein a gel is prepared and electric current is passed and the DNA flows through it. So what is the charge of DNA? It's positively charged or negatively charged? Anyone? Negatively charged, yes. So DNA is negatively charged, so naturally it will move towards positive charge. DNA has some mass, so if you have two fragments of DNA, both being negatively charged, both will move towards the positive point, but what will happen is, based on their mass, they will separate out. So this is basically a ladder that has gone in the gel electrophoresis gel. This ladder is a 50K ladder. So what I mean by 50K ladder is 50 base pair products are there, and the end band that is there is basically 50 base pair. The second product that is there is 100 base pair. The third product that is 150 base pair. The fourth product is 200, and similarly this darkest product over here is 500 base pair. I have calculated it. You can calculate it whether I am right. Common steps where things go wrong are your enzyme concentration is low. So you have to see during your standardization process at variable enzyme concentration what is your product. The second most important thing that goes wrong is the primer design. If the primer design is not efficient enough, the products will not be formed. So primer designing is one of the most important things in PCR that is there. Then GC-rich region. If the GC-rich region of the target region of interest is GC-rich, then again you will have variability. Sometimes you will get a product. Applications of PCRs are variable. Medical applications are genetic testing, prenatal testing, detection of mutations, diagnosis of disease, nowadays prognosis of disease, nowadays monitoring of disease, all happens on PCR-based techniques. Forensic applications, which were the most earliest applications wherein PCR was used, DNA fingerprinting, parental testing, Then research applications of sequencing, genetic mapping, gene expression, phylogenetic studies. These are broad applications of PCRs. Before going to different types of PCRs, I was just glorifying PCR to all levels, but PCR has its own limitations. You need to know them. Not many, but yeah, few limitations. Designing of the primers requires sequence info, so you will have to be very fluent in the informatics aspects and the biology of the disease or the gene that you are looking at. That is the most important thing. So you will have to understand products of PCR. One of the most important limitation that I have put at last is PCR is a sensitive procedure. Contaminations are bound to happen. So as I said, when I entered a molecular lab in TMH Mumbai first time, I had to wear shoe cover, I had to wear cap. I was given a new apron, which I was not even given during my MD pathology 3 years that I did. But for entering a molecular genetics, I was given a fresh apron. The aprons get washed on daily basis in a molecular genetics lab. I don't think even rest of the histopathology or hematpath people would be washing their aprons anywhere in 6 months. But molecular genetics lab, the reason these are some of the pieces that I myself have seen, and that is the reason I am showing it to you. Standard PCR is the routine PCR where you just amplify the product, and then that product can be used for multiple downstream applications or different applications, I would say. Allele specific PCR wherein the mutant good amount of physical infrastructure you have is not going to be helpful. The equipment, whether automation is required, consumables, standard operating procedures you will have to set in for each and every process, for keeping the simplest apparent to wearing cap, shoes. You will have to develop standard operating procedures, quality control measures. So purpose could be research, diagnosis. You will have to have quality controls for this purposes. This is the basic design. So whether it's your sample or reagents, all have to have a unidirectional flow from a pre-PCR room to post-PCR room. You cannot go the opposite way. Any which ways, if you go opposite, there is going to be a contamination. You need to maintain positive, negative pressures at separate rooms. There is sample reception room wherein the extraction happens. There are instrument room. There is template addition room. This is the most important room. Anything happens in this room, a smallest amount of contamination, anyway, it can go haywire. Then there are PCR setup rooms. These two are the sacred rooms. These are said to be the most important rooms. These are said to be rooms where if you get a contamination, that contamination can last for years. And you might start, regular basis you might get anything, non-specific bands or wrong results, or it might come up in between like a random error happening, but you will not be able to catch it. You need to maintain a unidirectional flow. That's the most important thing that is there. DNA RNA extraction room has to have its own specific infrastructure, equipment.
It's my pleasure to introduce our next speaker, Dr. Priyanki Amarabandu. Dr. Priyanki is a consultant histopathologist at Acacia National Institute of Cancer, Sri Lanka. She plays a leading role in establishing molecular pathology diagnostic services at Acacia Hospital, including the implementation of next generation sequencing. She graduated from the Faculty of Medicine, Colombo, and obtained MD histopathology from the Postgraduate Institute of Medicine, Colombo in 2007. Additionally, she completed a postgraduate certificate in clinical genetics.
Population 400,000 children and adolescents of 0 to 19 years all develop cancers. The most common type of childhood cancers in the world is leukemias, brain cancers, lymphomas and solid tumors such as glioblastoma and Wilms. Childhood cancer cannot generally be prevented. It's because or identified through the screening. Most childhood cancers can be cured with generic medicines and other forms of treatment, including surgery and radiotherapy. Treatment of childhood cancer can be cost effective in all income settings, whether it's high socioeconomic country or low, and actually it is an investment to a country treating pediatrics. This is. So we should let these children bloom and take over the country. The pediatric cancers at a glance in Sri Lanka 2020. Males, boys, 51% are leukemias, 11% bone tumors, 10% lymphomas, and 9 CNS and brain tumors. And when it comes to girls, 41% leukemias, 11% CNS and brain tumors, and 11% germ cell tumors, 7% bone tumors, and 7% basic utility. Potential for simple standardized procedures in auto-automation and rapid throughput. Increased number of techniques for infectious diseases and tumor diagnostics. Available reflex for equivocal morphology. Yes. The causes of childhood cancer are not yet clear. Unlike adult cancers, only a few environmental factors have been associated with cancer risk, including radiation, prior chemotherapy, parental exposure to pesticides, benzenes, polycyclic aromatic hydrocarbons, and smoking. Next generation sequencing has improved the understanding of the genetic basis of childhood cancer, while whole genome sequencing (WGS) is the most comprehensive technique that identifies variants in coding and non-coding regions. As I said, whole exome sequencing is a cost-effective approach, detecting up to 85% of disease-causing variants, and finally, targeted analysis sequences is much more long way for us to go. Focuses on a panel of genes known to have a strong association with specific predisposition, cancer predisposition syndromes beyond the verge of starting on diagnostic and precision. In Sri Lankan setups, starting molecular lab, we don't have any data, we don't have a biobank, we don't have any molecular mutation and birth analysis, so we don't know significant of an unknown variant. We don't know whether it is significant mutation with the diagnostic and therapeutic, whether we trying a new mutation unique to our population. And we don't get the feedback from the clinical teams. They get the wax blocks. They get the wax blocks, and they are not courteous enough for us to inform. They outsource the testing. They are not courteous enough to inform whether it is positive or not. But sad to say, sometimes the patients are very good and they come and give us an additional and funds expensive. We are struggling in the country. There are a lot.
Lunchtime.
But also to keep you awake after the lunch. I will try my best, try to be with me, because informatics is like, many of you might not have even thought about it, how it can be integrated into molecular. And it's a pretty new field, I would say. It's developed before NGS, it was Sanger sequencing, where the bioinformatic use was actually very pertinent and was important. Download some reference sequencing. If possible, I would show on live demo if the internet speed is good enough. Some sequence analysis software, mutation analysis, HGVS reporting. So HGVS is a nomenclature of reporting, the standard nomenclature of reporting a variant or mutation. And how it was used in a simple PCR. And now once we get a knowledge, or we get the idea of using this central dogma in molecular techniques, we use the raw data that comes out from this molecular techniques and use informatics to use the nucleotide protein databases softwares to detect mutations for diagnosis, prognosis, and therapeutic values. So coming back to center, there is something called as Galaxy website. You can go, it's from Broad Institute from US. They have developed a simple website wherein they have given on the left hand side multiple tools. The center there is a window, you just need to use different tools and see how the analysis can be done. That's for your practice, and with this I will stop. I will end this talk. And Mr. Priyankara, medical laboratory technologist, National Hospital Tangi, obtained his high diploma in medical laboratory technology 2007.
from the Medical Laboratory Training School, India, and Bachelor's of Medical Laboratory Sciences Honours degree from the Open University Sri Lanka in 2021. He also hold an executive diploma in service management obtained from the Open University. His services during the COVID pandemic is commendable, and during this period he mastered the PCR techniques of the virology laboratory. I invite Mr. Priyanka to outline the process in our molecular laboratory.
Good afternoon, all of you. I would like to thank you. I am Priyanka. I am working in the molecular diagnostic lab in this hospital. This is one of my capture of our